The story of protein synthesis takes place in two chapters. The first is called transcription, and the second is called translation. Each takes place in a different part of the cell and is controlled by a different enzyme. Now, to understand protein synthesis, it's helpful to use an analogy at first. So we're going to use an analogy of someone that has a cookbook and someone that can do the cooking. So, the someone that has a cookbook is named RP, which stands for RNA polymerase. RP possesses a cookbook that contains all of the recipes that a cell would ever want to make. But this cookbook is precious, and therefore it is required to stay in the nucleus at all times. Now, RP decides that it would be good to have some chocolate chip cookies around. So, since the cookbook can't leave the nucleus, and the kitchen is out here where the ribosomes are, remember the ribosomes are the structures that actually make proteins, the way that RP is going to get these chocolate chip cookies made is to first make a copy of the recipe onto an index card. Index cards don't last very long, but they don't need to because they're not the originals. And the index card, which in this case is mRNA, is able to leave the nucleus. All right? So RP has a cookbook with all the recipes or all the genes. The gene that it wants to make into a protein gets transcribed or rewritten onto an index card. Now, this index card or mRNA can leave the nucleus and go into the kitchen, which is the rough ER, or wherever the ribosomes are. Now, the ribosome is the next enzyme, and it acts as the chef. It's able to read the recipe on the index card or the mRNA, and from those instructions, actually make a protein or chocolate chip cookies, whichever way you want to think about it. So, transcription, number one, takes place in the nucleus and is where a gene that is on the DNA is transcribed into the gene on mRNA. And this is done by RP or RNA polymerase. Now, the analogy is that we have a recipe in a cookbook and that's transcribed or rewritten into a recipe on an index card, sort of the way you might think of a librarian doing something that belongs in the reference section. The second chapter, translation, takes place wherever there is a ribosome, whether that's in the rough ER or out in the cytoplasm. And the ribosome takes the recipe on mRNA and translates it into a protein. And in the case of our analogy, this is like reading the recipe on the index card, where the chef reads the recipe on the index card and ends up making cookies.